Our next speaker is Christina Yen. She serves as Senior Manager of the In Vitro Release Testing Department at Targus Pharma. She leads a team of 8 analysts for development and validation of IVRT methods to support generic drug development and post-approval manufacturing changes in compliance with SUPAC-SS guidance.
Hello, everyone. I just want to make sure to know that you can see my presentation okay?
You either need to share the other screen or duplicate your slides.
Okay.
What you can do is you can go up to display settings. Oh, there you go.
Oh, that's fine. Thank you.
Good?
Yes.
Thank you. So my talk today will focus on the method development activities for IVRT methods that support topical product development. I'm Christina Yan, and I am the Senior Manager of IVRT, which is part of the In Vitro Sciences Department, along with IVPT and Skin Biology at Targus Pharma. Together, we have amassed a lot of experience as an end-to-end contract service provider for topical pharmaceutical products. So I'm going to walk through an organized approach to analytical and IVRT method development and share some recommendations on studies and technique. I will be presenting studies in a sequential order. It's important to note that the analytical and IVRT methods are for the most part developed concurrently and that they often inform each other of any changes that may require further method optimization of one or both of the methods. We typically look for an existing method as the starting point for analytical or HPLC method. A method can come from the USB monograph. It could be one provided by a sponsor or one found in the literature. The method is then adapted, among other things, to have much shorter runtimes. So short runtimes are important because of the large number of samples generated during IVRT studies. As an example, for a study containing 132 injections, a 15-minute run would take 33 hours to analyze. That can be significantly reduced if the runtime can take about 8 minutes per injection. If there is no method available, then we develop it from the ground up. In both cases, it's important to have a good understanding of the physiochemical properties of the active and of the semi-solid dosage form. It is very important to understand solubility, for example. So the end product should be a method that has a well-resolved peak, has a good capacity factor, is sensitive, precise, can analyze a range of concentrations, has no interference of the active, and is robust to small variations such as mobile phase composition, column temperature, and the temperature of the lab. We initiate our development by establishing preliminary analytical method conditions that will then support IVRT method development activities. If developing the method from the ground up, this means screening mobile phase and columns, evaluating flow, column temperature, and other settings. Initially, we use a PDA detector to determine an appropriate wavelength. I'll circle back around to the key studies that are performed for the analytical method development. But for now, once we have preliminary HPLC method conditions, we shift our focus and begin to develop the IVRT method. So again, the basic principle here is that there should be passive diffusion of the drug from the product matrix into the receptor medium to produce a steady state release rate that can then be used as a comparison tool. The IVRT method needs to be precise and linear. It needs to have a pseudo-infinite dose, and it should use a membrane that does not bind to the active. To achieve this, the following studies are performed. At the start, 2 runs of 3 receptor media and 2 membranes each are performed typically back to back. Receptor media candidates are aqueous or aqueous organic solutions that take into account the solubility and other properties of the molecule and the type of formulation that it's in. During this run, observations are made to evaluate membrane integrity. Things like discoloration, curling, or sagging of the membrane, for example, could indicate that the membrane may not be a good candidate. Membrane binding is assessed for all the membranes that were screened in the first study and any additional ones if needed. A standard solution is used for this activity. To establish linear release, we perform a up to 24-hour run with about 8 sampling time points with the goal of getting to at least 70% dose released just for this experiment. This is in order to evaluate the release curve and to make sure that we have sampling time points in a section where there is steady state kinetics and that there is no lagging or plateauing. So by now, hopefully, the following may have already been identified. So 2 lead receptor media, 1 lead membrane typically, the sampling time points and the duration of the run, along with other more standard conditions, right? So like sampling volume, for example. If that is the case, then a screening study can be performed for each of the 2 leading receptor media to evaluate their ability to be selective, sensitive, and specific to changes in the API strength of different formulations. This is carried out using 3 cells for each of the formulations. And the formulations that are being used are a nominal, one that is 50% rather of the nominal, and one that is 150% of the nominal. A lead receptor media is then established and the study is repeated, this time using 6 cells per formulation. Another key study is selectivity for an intentionally altered formulation. So examples of changes are the type or amount of an excipient or a key process of manufacturing. These are all the core studies, the core method studies. There are other additional studies that are performed in preparation for validation. For example, robustness assessments, inter-day and inter-analyst intermediate precision. I won't cover this here because that's described in other validation presentations. So here's an example of the data for specificity, selectivity, and sensitivity of 3 different strengths, the nominal, a 50%, and a 150% of nominal. The first figure demonstrates the different release rates for different strengths. Specificity demonstrates the proportionality of release for the 3 formulations. Selectivity demonstrates the 3 formulations, that the 3 formulations are statistically different. And lastly, sensitivity provides a measured percentage for the lower and the higher strengths. For every method, we evaluate the 2 vertical diffusion cell types that we have in-house. Both are for manual and dosing and sampling. The apparatus on the left holds a larger amount of receptor media and is dosed by spreading the formulation with a spatula onto the membrane in an area created by a Teflon dosing wafer. The surface area of this membrane is also greater. The apparatus on the right holds less receptor media and is dosed by transferring the formulation from a syringe onto the membrane in an area created by a glass donor chamber. The tip of the syringe has been cut off to try to reduce the effects of shearing. The surface area of the membrane for this vertical diffusion cell is smaller. In both cases, the formulation is occluded throughout the run to protect it from any changes due to evaporation. The sampling stems are also occluded to prevent evaporation of the receptor solution. And this graph demonstrates that different apparatus result in different steady state release rates. So this provides some flexibility when trying to establish good release rates. If neither of the vertical diffusion cells yield an acceptable method, then we look into our immersion cells on apparatus 2. Once we have established the conditions of the IVRT method and know what the lowest and the highest expected sample concentrations are, we return to the analytical method, since we now can prepare simulated samples or QC samples at various concentrations using placebo receptor solution. With these QC samples, we can study the accuracy and precision of our method, which is a critical parameter. We also assess the specificity of the method, as well as linearity, range, and LOQ. We assess the stability of the sample in standard solutions at ambient conditions, exposed and protected from light, and at refrigerated conditions, so in the range of 2 to 8 degrees. We typically analyze the solutions at about 24 and 48 hours at a minimum. We also perform a study to evaluate the stability of the sample throughout the run. So to do this, we place a high and a low QC sample in a water bath at IVRT method temperature for the length of the run. We compare it to high and low QC samples that have been stored at ambient conditions for the length of the run. Then we perform an additional solubility study. In this case, we use the final receptor medium at the IVRT temperature, so either 32 or 37 degrees. It's important that we ensure a solubility of various folds higher than our highest expected sample concentration. So we try to have at least 10 times the solubility of the highest expected sample concentration. We have found that performing a long run, maybe 20 or 30 minutes long with the placebo receiving solution, helps to see if there are any late eluting peaks that are coming from any excipients in the formulations that may cause interference if they elute in subsequent runs. If using a buffer, it's a good idea to perform a quick crash test with any organics that are being considered for use. Once the type of column has been chosen, it's a typically good practice to perform the studies with newly acquired columns to make sure the final decisions are being made with the most reliable columns in preparation for validation. Good preventative maintenance for the vertical diffusion cells is essential. So because actives can stick to the glass, buffers and buffer salts can accumulate around the cells, organics and acids can corrode the cell holders, and algae can grow in the water jackets and the tubing, all of which can cause problems. Making thorough observations during setup, dosing, sampling, and disassembly can provide support for the empirical data, and also can help identify any issues such as bubbles in the receptor solution, back diffusion into the donor chamber, or any formulation changes over the run. Also, IVRT is very technique-based, so uniformity of training is important. So we will now look at a few technique-driven aspects of IVRT testing. So it's important that the IVRT conditions are very well controlled. In this example, membrane temperature is being measured for both of our vertical diffusion cell types. The exact distance and position from the membrane are taken into account when using the infrared thermometer to ensure both accurate and repeatable measurements. So dosing is demonstrated here by transferring an aliquot of product onto the membrane and then carefully spreading it with a spatula, trying to prevent any voids between the formulation and the membrane. A glass disc is used to occlude the formulation. And the picture on the right illustrates dosing onto the donor chamber with a syringe. Applying even pressure and dosing at an even pace is important to help reduce the effects of shear. During manual sampling, it's important to use a calibrator or pipetter to draw the sample from the middle of the vessel and to perform the same actions across all the cells. With manual systems, staggering dosing and sampling ensures that the time elapsed between time points is the same across all the replicate cells. Again, making observations throughout the experiment is key because, for example, any changes in the membrane receptor solution or product appearance could indicate possible issues. So as I wrap up, I'd like to acknowledge my team members listed here in alphabetical order. They are dedicated to producing great methods and are always looking for ways to advance our processes, and they make work fun. Thank you, everyone.